everyone. Thank you for being with us. I am Bratati and welcome all of you to Bratati Science Class, your favorite science channel. Short explanations, easy explanation, easy to remember. So let's begin class 9, chapter number 5, and which is the fundamental unit of life. Okay, so what am I talking about? Fundamental is basic unit of life. Like what is the smallest part of us? Can you tell me? You are absolutely correct. We call it cell. So we are diving into cell today. We are going to see what is there inside the cell. C-E-L-L, not this cell, but C-E-L-L, cell. That is the smallest part of any living organism. Okay. So let's start. I will be sharing my screen. All right. I hope it is visible to you. So this is the picture of cell. Okay. We are going to see in detail. So let's move to what is cell. As I said, it's a basic structural and functional unit of life. Structural, that is, you consider it's like a brick of a building, right? So if we are seeing a brick, from one brick, we cannot make a whole building. We need many cells or many bricks to be together with the help of cement, sand, and other things to make it a building. Functional again. So, like we have the brain, which is the control center of our body. Similarly, the cell has inside a small control center, which is called nucleus. That we are going to learn about it in future. But the cell is the functional unit of life. So, ready to dive into it? Structural organization of the living organism. Repeated, it's living organisms. Okay. So as I said, compare the cell with the brick. Okay. Many cells, they have to be together with the help of cement and all to make one tissue. Right. So many cells are put together to build one tissue. Now tissue you can compare with one wall of a building. It's just one wall. Now like this, we need four walls to be together to make a room. Four walls, a roof, that will make one roof. Now these tissues together, few tissues together, they will prepare the organ. Now cell, as I said, cell can be the blood cells, it can be the muscle cells, and together they are forming the muscle tissues, right? The tissues together forming an organ. Now, how can we compare an organ? We just prepared with the wall, four walls or five walls, including the roof. They're making a room. So we are comparing the organ with the room. Okay. Now, many rooms together, like we have dining room, we have bedroom, we have kitchen. So different rooms together make a complete apartment, a flat, what we can say. So this apartment we are comparing with our organ system. So many organs together, they form an organ system. Organ system, for example, human, we have digestive system, respiratory system, circulatory system, respiratory system, reproductive system, and so on. So different organs make one organ system, which we are comparing with the apartment. Now, many apartments are there together, which are forming a gated community. So finally, all these kind of organ system, digestive, respiratory, reproductive, circulatory, excretory, together they are forming a human body. So we are comparing the organism. Finally they are making an organism, and this we are comparing with a gated community. I hope this is an easy example that you can remember well. So cell, that is the building block of the body, which we have compared with the brick. Several bricks together will make a wall, which we are comparing with the tissues. Several walls, 4 or 5 walls together will make a room, which we are comparing with the organ. Different rooms together will make the organ system, or it's a compare with the apartment. Many apartments are there together, which will make a gated community, and finally we are making the organism. Okay. Now we are moving when it was discovered and by whom. It was 1665 when Robert Hooke took a bark of a tree, from the stem. He cut a part and looked under the microscope where he could see dead cell. So he coined the term cell because he could see some kind of this honeycomb structure. And so as it looks like a cork, so he gave the coined the term as cell. Okay, so he is the one. If the question comes who coined or who discovered cell, no doubt the answer is Robert Hooke. Okay, but who is the one who discovered the living cells for the first time? Then the answer is Antony van Leeuwenhoek at the year 1674 when he discovered a living cell for the first time. After many years, you can see the year is 1831, came Robert Brown, who discovered the control center of a cell, that is the brain of the cell, which is nucleus. Then in 1839, Parkinson coined the term protoplasm, and protoplasm is the combination of the nucleus along with the cytoplasm. Together, it's a jelly-like substance. Okay, so this is clear, the discovery part. You're moving next to the theory. The cell theory here, Sladen, remember the name. Sladen is a botanist. Botanists are those who studies about plants. Schwann is a zoologist. Zoologists are those who studies about animals. So this botanist and zoologist together, they said, in this world, all the plants and the animals, they are made up of cells. This is called as cell theory because they said all the living organisms basically are made up of cell, and this is termed as or coined as cell theory. Now we are moving to the classification of cells. The types of cells basically depend on the number of cells present in an organism. So there are few organisms which have only one cell. One cell organisms are called unicellular organism. For example, amoeba, paramecium, bacteria, euglena. Here again we will be learning about prokaryotic cell and eukaryotic cell. In this, amoeba, paramecium, bacteria, they come in prokaryotic category, and euglena comes under eukaryotic category. Okay, eukaryotic cells generally have very clear nucleus, that is the control center, but prokaryotic cells lack the nucleus. Rather, they have nucleoid. We will know this term in future. Soon we are coming. Okay, so hold on tight your seat and get ready for the next thing. We are knowing for multicellular. The few organisms, they are made of more than one cells. For example, we humans and all the plants and animals, they are made up of many cells. So they are called multicellular. Okay. Now, children, you may think, or audience, you may think, why I'm going so fast? The reason behind it, in book, it's given in details and you get many videos of a lengthy lecture and all. But I here tried to make it short and like a summary for you, or you can call it as last minute revision. So tomorrow is the exam and I have to revise the chapter, or I'm preparing it for 7 days. You need a nutshell, and that too should be in short time. So that keeping in mind, I have prepared these slides and I'm explaining to you like a quick revision. You know all the things, but you need a quick revision. Okay. The cells are of different shapes as well as different sizes. All cells in the world, they are not close to resembles to each other. They have the different shapes. For example, the spiral shape. Okay, or it can be spherical like ovum, egg cell, that is spherical in shape. Rod-shaped bacteria, it looks like a rod, like bacillus. Okay, comma-shaped bacteria are also available, which are called Vibrio cholerae, cholera's, bacteria, that is a comma-shaped one. Spindle-shaped is like this, that the end part are sharp and inside there is a swollen one. This is example of the muscle cell. Okay, so spiral, spherical, rod-shaped, comma, spindle like muscle cells, also branched like the neuron or the brain cells. These are the branched kind of thing. So this is branched. So different shapes of cells and the size of the cells are also different. Smallest cell till now it is mycoplasma bacterial cells. So the smallest cell, if the question comes 1 mark, answer is mycoplasma species. So write species because the mycoplasma is the general name and then we can write species. Actually it is Ledloi, but no need to write, you can write SP for species. The largest is egg of ostrich. The ostrich, the bird, the egg of ostrich is the largest cell. A single cell, yes. The egg shell, that is a single cell and it's the largest cell till that. Now we are moving to what is present inside the cell. I'm going to tell you the details in the next slide. But here, just let you know, cell wall, that is the outermost part of the cell and which is present only in the plant cell and bacterial cell or fungi cell. So cell wall is a rigid structure, okay, and is present in plants cell, which we call cellulose. It is present in bacterial cell, which we call it peptidoglycan. And also it's present in the fungi, which is called chitin. So it's a cell wall of fungi, which is called as chitin. Cell wall is not present in animal cells. Okay, again I'm repeating, it's not present in animal cells or it's not present in eukaryotic cells. Cell membrane, this cell membrane is a permeable membrane, which is called its selective thing, selectable, because selective movement of the substance from and towards inside or outside from the cell. Okay, so cell membrane is also called as plasma membrane. Cell membrane, or we can also call it as plasma membrane, right? Which is selectable, permeable, selective, permeable. Only selective things are passing through it. Next is the nucleus. So we talk about cell wall, we talk about cell membrane. Nucleus is the control center of the cell, or we can call it the brain of cell. Remember, cytoplasm, sorry, here no much place. Another slide, I could keep it for this. Cytoplasm is the jelly-like substance that is present in the cell and which holds all these cell organelles. Cell organelles are the different parts, the membrane bound parts, okay, that are present in the cytoplasm. So we have discussed all the components in short. Cell wall, which is present only in the bacterial cells or plant cells or fungi cells, but not in the animal cells or eukaryotic cells. It is very rigid and dead in nature. Next comes the cell membrane, which is present in all kind of living organisms, which is selectively permeable because it selects some of the substance to pass to and from the cell and inside the cell. Cell membrane also has the name called plasma membrane. Nucleus is the brain or the control center of the cell. Cytoplasm is the jelly-like substance present in the cell which holds the membrane-bound cell organelles. Clear? You're moving to the next. Now here I'm going to show the cell. We have taken a spherical shaped cell, but actually it is not identical. As I told you, the shape can be like this or it can be like this. All the shapes, all the cells are have different sizes and shapes. They don't look identical sphere-like structure. Okay, done. Now, a very big nucleus. Then we have the endoplasmic reticulum. This is the Golgi apparatus. Outside for plant cell and bacterial cell, there will be the cell wall. And inside the cell membrane, which is a bilayer of phospholipid, lipid, protein, lipid. So cell membrane is the bilayer. Two layers are present here. We have plastid, chromoplastids. We have mitochondria, that is the powerhouse, and these are the finger-like structure which are called the cristae. Lysosome is present. The rest of the part is the jelly-like substance. As you know, this is the cytoplasm. So basically, if we do labeling in tools, okay, so this is the nucleus. This is the ER or endoplasmic reticulum. This is plastid, which is present only in the plant cell. This is the mitochondria, which is the powerhouse of cell. This is the Golgi apparatus, which is the having it is basically that it helps in the transportation in the cell. I will tell you. And this is the lysosome. Lysosome is sometimes called lysosome, suicidal bag, or it has the digestive enzyme that digest all the things. So this is the 2D structures of the Golgi. This is mitochondria as the powerhouse that generates the ATP or for the energy. This is ER, endoplasmic reticulum, and this is the control center or nucleus. So, and this is the cell wall. This is the cell wall which is present, as you know, in the plant cell, and this is a bilayer cell membrane, or we can also call it as plasma membrane. This is clear. So if in question it comes to draw the diagram of the cell, you can draw this is a cell and these organelles are present, cytoplasm is present, nucleus is present in it. Inside the nucleus we'll go in details, like there will be nucleolus, there will be the DNA, there will be the nucleoplasm. So that we will come in the next slide. So this is cell. Now cell wall, as I told you, I want this. Cell wall is rigid, okay, dead, outer covering, which decides the shape of the cell, which is present in plants, as I said to you, in the form of cellulose, bacteria in the form of peptidoglycan, and in the fungi, which in the form of chitin. Now, cell membrane, as I said to you, it is a flexible, which is protein, lipid, LPL, and this is how they form. Bilayer, so lipid, protein, lipid. This is how the solid membrane is formed, and membrane is a semi-permeable in nature, which selectively helps in transportations or the movement of the solute or the solvent, okay, movement of the solvent, only solvent from outside or inside, okay. Now, they help in transportation, as I said. The transportations can be of active and passive. The passive transportation, there are the two types, which is the diffusion and the osmosis. In active transportation, the solvent is the water. Only the solvent molecule passes through, not the solute. The solvent passes through the semi-permeable membrane, that is the plasma membrane. And in passive transportation, it's always from high to low. And from active, it's just the opposite. The solvent moves from low concentration to high concentration. So what is this I'm talking about? I'm talking about the concentration of the solvent, right? So this is concentrations. Passive transport, the solvent moves from high to low concentration. No further energy is needed from outside. But active transportation, they need a lot of energy, which is called ATP, where ATP stands for adenosine triphosphate. Now, passive transportation is very slow movement for the small molecules which can move without any extra energy. But in active transport, the very large molecules that move very rapidly, and that's why they need the energy from outside. Okay, done. So now we are going to learn about what are the different types of solutions present in the cell. Hypertonic solutions. Name it hypertonic. If it is hypertonic, that means this is the cell. Inside, this is the nucleus, and inside the solute is more in number compared to the solvent. If the solute is more, solvent is less, the concentration is more inside compared to the outside. So there are few solvent and inside there are more solvent. So here it is high and outside it's low. So what will the solvent will go from high to low. So high to low, this movement, the movement from high to low. Why the color is not changed? Okay, let me take green color. So high to low, this movement, the solvent will come out and the cell will shrink. The big cell will shrink. So how to remember? We have R here, R, and here also we have R. So hypertonic solution means the cell will shrink. The question comes, if you put a cell in the hypertonic solution, what will be the result? Whether it will swell or it will shrink. The answer is it will shrink because they will show exosmosis. Exosmosis means from high to low, the solvent will come out from the cell to the outside. Clear? Now moving to the next one, which is hypotonic solution. There is a big O. That means it is the swelling of the cell. What happens here? Again, this is the cell and this is the nucleus. Inside the concentration is less, but outside the concentration is more. So this is high outside and inside it's the low. So where this solvent will go from high to low, from outside, lot of solvent or water will go inside to make it swelling, and this is called endosmosis. So clear how to remember? Shrink R, hypertonic R, hypotonic a big O, that means the cell will swell, right? Another kind of solution can be isotonic, where outside and inside the cell, outside and inside the same concentration, so no movement of the solvent. Those kind of solutions are called isotonic, right? Okay. So now, as we said, active transport, that is from low to high. Active transports are of two types again, endo and exo. Remember, endo means something is coming inside. So amoeba, we already told it is the single cell organism amoeba. When they get the food from outside, they have a vesicle, and with that they just engulf the food inside. So food is engulfed by the amoeba by the process of endocytosis. That is again active transport. So you know it needs the ATP, the energy. That ATP is generated in mitochondria, okay, in the cell. Now exocytosis, exo is something is going out. So what do the cell throw out? It's generally the waste material. So whatever waste is present, they form a vesicle and they throw it out of the cell, and that is the cell shift the waste to the extracellular space, that is the outside of the cell, which is exocytosis. Okay. Now plasmolysis. Plasmolysis is the shrinkage of the plant protoplasm. Protoplasm is again the jelly-like substance that is present in the plant cell. Okay, and then it is moving away because again it is put in the hypertonic solution. That means the protoplasm or the cytoplasm is getting shrink, and so they are moving away from the cell wall. then when they are placed in the hypertonic solution. So whenever R is there, remember there will be the shrinkage, and then the cell will move out away from the cell wall. Clear? So cell membrane or the protoplasm part will move out. This is the cell wall, and the cell membrane will come and shrink to this. Initially they were like this much, and then they are shrink to this. Okay. So this is clear. Active transport is clear. Now moving to the organelle, which is the nucleus. I will take, which color should I take? Nucleus, it has like this, a pore in between. Though it is spherical in shape generally, but it's not complete. There are the pores in between. Then there is the nucleolus. And then, as I said, there is the genetic material present here. And the rest of the part, again, the jelly-like substance. Now here, if we mark, this is called the nucleolus. Inside the nucleus, again, the concentrated part is the nucleolus. These are the nucleopore. Pore means the, obviously, it is the holes inside. This is the nuclear membrane. Nuclear membrane. Then this is the DNA, this is the genetic material DNA, which decides what will be the color of our eyes, how will you look like, okay. And that's why due to DNA or our genetic material, we looks very similar to our parents. So this is about the nucleus, which is very important, the control center of cell. Okay, so nucleus decides like what to do, and these are the parts of nucleus. Now this DNA, during the cell division, the cell breaks, and from the mother cell the daughter cells are produced. So then when the DNA is tightly coiled and condensed, so they become like tightly coiled and condensed, then they produce this chromosome. So here this is the nucleus. Again, this is the nucleus. These two parts are called chromatid. This is a chromatid and this is a chromatid, and together they call the chromosome. So during division, there will be different form like this, and all this we will learn. So as of now, know that chromosome is the genetic material part, which is the have the chromatids and have the DNA inside, which decides the our specialist how we will look like, the color of the eyes, and so. Now, based on the nucleus, the number of the nucleus, or the based on the nucleus, the types of cell. We did it before the prokaryotic cell. We did it before the unicellular and multicellular. So now types of cell can be the prokaryotic cell and the eukaryotic cell. So prokaryotic cell, actually they look like this, and they have flagella. This is a moving part, and they don't have a clear nucleus. As I was telling you, they just have a nuclear material present like this, which is called nucleoid. They have the cytoplasm. They lack the cell wall. They have the cell membrane. They lack the cell wall, flagella, and they have the plasma membrane, and they have ribosome. So which are the parts they have? This is called the nucleoid, the genetic material. They have the plasma membrane or the cell membrane for selectively taking the solvent. They have the ribosome, and this ribosome helps in synthesis of the protein, and these are the flagella that helps in the movement of the prokaryotic cell. So this is the structure of the prokaryotic cell. Now eukaryotic cell, as we know, the animal cell, the plant cell, these are the probably the proper cell wall. Animal cell do not have the cell wall. They have the proper cell membrane. They have the proper nucleus. So prokaryotic cell and eukaryotic cell, the main difference is prokaryotic cell does not have a nucleus, but eukaryotic cell has a nucleus. Okay, the prokaryotic cell has flagella for the movement, but eukaryotic cell do not have a flagella. The prokaryotic cell do not have a cell wall at all, but eukaryotic cell, plant cell, have the cell wall. Got it? Now, cell organelle. The cell organelles are of single membrane, double membrane, and membraneless. In single membrane, we have the vacuoles. We have Golgi body, It is also called as Golgi apparatus. We have ER, which is the endoplasmic reticulum and the lysosome. So these are single membrane organelles. The double membrane organelles can be the nucleus. This eraser. Why this is not coming? Eraser. Okay. So double membrane can be nucleus, nucleus, and then nucleus, mitochondria, okay, and plastid. Membraneless can be ribosome and centrosome. So these are the organelles which do not have the membrane. These are all present in the cell. Now this is the structure of the endoplasmic reticulum. This is the 3D structure. This is our nucleus. And there this is the ER, endoplasmic reticulum. It can be smooth ER and it can be rough ER. In rough ER actually there are ribosomes present. That's why rough. Ribosome ER. Smooth ER, no ribosome. Okay, ribosome is not present in the smooth ER. Rough ER, that as the ribosome is present, and you know the main function of ribosome is synthesis of protein. So this rough ER can synthesize protein. So protein synthesis. So they synthesize protein, and smooth ER, they synthesize lipid. So they synthesize lipid is the fat. So they synthesize lipid. Smooth ER, they synthesize lipid, and this is the rough ER which synthesize the protein. Okay. Now Golgi body, the work for Golgi body is they look like this. This is the Golgi apparatus 2D shape. Now they are mainly they modify the protein. So and they help in the transportation. So what do? They do like packers and movers. Okay, what do? They receive a parcel, generally packers and Agarwal packers and movers, or this DTDCs and all. What do? They receive a parcel, and after they do the packaging and all, then they send it or distribute into the respective addresses. So here Golgi body, once from the ER, from the endoplasmic reticulum, when they are getting the protein, they modify the protein to some extent with some folding and all. So once the protein is modified, then they store it for some time. Then they start doing the packaging, and through the packaging, they share it and transport it to the other organelle. So they basically do this transportation. So Golgi body, the main act is the transportation of protein to different organelle, right? So how they do it, I told you already. And lysosome, which is also known as the suicidal bag, that is present in the cell. Actually they have many digestive system present in the body, helps in the digestion of different cells and different enzymes. And those not system, actually they have many digestive enzymes. Black ink, digestive enzymes, and sometimes they eat their own cells. Okay, and then they help in digesting the waste material also. Next is the vacuole. In the plant cell, the vacuole are very big in size, but in the animal cells, some of the animal cells, they lack the vacuole. What is the vacuole? Again, the cell membrane of the vacuole is made up of tonoplast, and they basically store the water, minerals, and food. So basically what I will write, they store water, food, minerals. Okay, the plant vacuoles are very big, gigantic, but few animals, they do not have vacuole, and the membrane of the vacuole is made up of tonoplast. Okay. Now plastid is present only in the plants. Present only in plants, not in animals. The plastids can be chromoplast, which is required for to detect the color of flowers and fruits. Colors of flower plus fruits. There can be chloroplast, and the chloroplast is for the green color pigment for leaves. And there can be leucoplast, that is the colorless pigment, and which is actually for, the roots are colorless, so leucoplast is responsible for that, the color of root. Mitochondria, as I told you, mitochondria, they have, this is the structure, see the finger like structures are called the cristae, and they can produce the ATP inside through ATP production through the aerobic respiration, and this is called the powerhouse of cell. Okay, so because they produce the generate the ATP, and the ATP is produced through aerobic respiration. Audience, please consider my handwriting as I'm using the pen and the tab. Okay, so that's why the handwriting is like this. Okay, so ribosome. The ribosome, as I told you, it's a protein factory. They synthesize the protein. Okay, and they are present in bacterial cytoplasm. As we told you, prokaryotic cell, they have the ribosome. They are also present in the eukaryotic cell, RAF-ER, the RAF endoplasmic reticulum in cytoplasm, and also in mitochondria. So ribosome is present in the various organelles and also in the chloroplast. So ribosome, what we call, the special name is they are the protein factory of cell. So see, all one mark question will come. The powerhouse of the cell is, answer is mitochondria. The protein factory of cell, the answer is ribosome. The control center of the cell is nucleus. So this is how the suicidal bag of cell is lysosome. The packaging system or transportation, who helps in transportation, the answer is Golgi body. Then ER has two functions. One is the productions of lipid, that is ACR, and the productions of protein by RER. Clear? So protein factory, where are they present? They present in bacterial cytoplasm. They also present in eukaryotic cytoplasm, comma RAF-ER, even mitochondria. So as I said, it's a protein factory. Now cell division, the cell can be divided in two processes. One is called mitosis, another one is called meiosis. So mitosis is basically from one cell when two daughter cells are produced. Meiosis when the mother cell, four daughter cells are produced. So this is the main basic difference also. In mitosis, if the chromosome number is 6 of mother cell, the daughters also will have the same number of chromosome. But for meiosis, if the chromosome number is 6, the daughters will get 3 chromosomes each. So they are getting half of the chromosome from the mother. So what is the basic difference? The first difference of mitosis and meiosis is, we can say, it is equational division. This is equational division, and this is reductional division. Number 2, same number of chromosomes, but it is half number of chromosomes where in the daughter cells. In daughter cells. Okay. Number 3, that is two daughter cells are formed and here 4. 4 daughter cells are formed. Number 4, mitosis helped in the growth and repair of cells, but meiosis helps in the generation of sperm cells, productions of the gametes, sperm cells or egg cell. So production of gametes, gametes can be of sperm or eggshell, the ovum. Again, this eraser, not this eggshell. Okay, so we are done. Quick recap. So what we have decided, what we have done today, we will stop sharing. I hope you liked today's session, dear. So it's all about like cell, what is inside the cell. We today dive into it, the fundamental, structural and functional unit of life. We know about the cell wall, and then inside the organelles like cell membrane, cytoplasm and different organelles. We know the short form of it. And if you like any part of the video, please click on the like button, and subscription is free. Please share the video with your friends so that everybody can see in the corner of any part of the world. I would like that if you comment something. Okay, so see you in the next video.